thank you for taking the time to come here to listen to my talk. And I will try to make it a little bit accessible. As I stand here to share with you, I am deeply honored to be able to come to this city where I grew up. So, I was born and raised in Macau, as Dean Chen said, in a very humble family. The reason is humble because my father gambled away all his money in Macau, which is not a bad thing for us. And my mom almost single-handedly, with my grandmother, raised the whole family. There were 5 of us. There are 5 of us. And I am this one, the dumbest looking, and the luckiest because of the support of my brothers and sisters that I managed to go to college. So I study in Yue Hua College, and it gives me good training, allowing me, this is just showing, to take A-level exams and so on to let me get into college. But I was never a really good student. I could do okay. This is my transcript. It may be difficult to see. See, I'm only getting points like 60s, 70s, 80s at the most. I think most of you are getting 90s. If you get 95 points, I think many of you would be very upset. I thought if I were to apply to University of Macau today, I very much doubt that I would be able to get in. So I am very honored and glad that I have this opportunity to be associated with this wonderful university. Thank you. Unfortunately, my mom passed away during COVID. This was a picture taken about 12, 13 years ago. These are my brothers and my sisters. And this is a picture that was just taken the past summer, from the oldest to the youngest. Again, I was the youngest and was the luckiest. My nephew, wife, sister-in-law, and my niece is really the support of my family. As I stand here to deliver this lecture, it's not about my accomplishment, but it's really a testament to the sacrifices and the unwavering support my family has given me throughout the years. This lecture is dedicated to them. So let me give a very brief background of Alzheimer's disease. It sometimes also goes with the Chinese name of age-related dementia. It is true that it's mostly associated with people 50 years or older, but there are cases, although very rare, that young people can also suffer from this disease. The important thing is that although, for example, significant research has lowered the morbidity rate of cancer in the past 2 decades, but the death from AD have continued to rise and will only get worse. And it's a significant financial burden to the society. And so this is a significant, tremendous healthcare problem. This is some of the statistics for people over the age of 65 and 75, 85, and so on. The important thing is that this will double by the year of 2060. So something needs to be done. There is still currently really no effective treatment. That's because the pathogenesis of AD is complex, and even after decades of research, the pathogenesis is still not fully understood right now. It is most likely a combination of genetic, environmental, and other factors contributing to the risk to the progression of AD. Unfortunately, it's an irreversible neurodegenerative disorder. There's really no cure right now. One could only see that there are two hallmark pathologies that would characterize AD. And one is the amyloid beta plaques and the neurofibrillary tangles. And both of these would lead to neuronal death and synaptic loss in the neural cells, which in turn would cause inflammation in the brain. So just very quickly, these are the, as I was saying, hallmark pathologies. One would see these plaques. These are insoluble mass accumulating in the brain. They aggregate and they disrupt the cell-cell communication in the brain. Neurofibrillary tangles are caused by abnormal tau protein. They also clump together to form tangles inside the neurons, hindering the neuronal transport system, mostly affecting the microtubule system and causing cell death. So the current drug treatment of AD, at least in the US, and I believe is throughout the world, there are only 7 drugs that are clinically approved. These are the drugs that have been actually just approved this year. And these are monoclonal antibodies to stimulate the immune system to break down the plaques. And is given by intravenous infusion. And this one has to be given once every 2 weeks, and this one has to be given once every 4 weeks. These 2 drugs are approved only for the early stage AD patients right now. And it shows clinical improvement, but it is mostly to arrest the progression. And it has been also shown to some improvement in the functional activity. The other 5 drugs I listed here, they are only mitigating the symptoms. They are meant to increase the number of neurotransmitters in the brain so that could improve the neural functions. These drugs do not address the underlying cause of the disease. But this is all that are available right now. In the field, people have been trying hard to look for other ways to tackle this problem. So people have been trying gene therapies that may be able to address the underlying cause a little bit better. The gene therapy approach would be, for example, to target the risk genes. And so these are 2 of the more common genes that people would like to target. People with these 2 genes have a significantly higher risk of developing AD. Some would target the genes that produce the amyloid precursor protein, and some would also target the gene that is associated with the tau protein. And so these are still in the really preclinical stage. They are experimental therapies. It still might take a long time, but there is a lot of effort going on in the field to try to target the underlying genetic causes. In the past 4 to 5 years, it emerged that the ApoE4 gene is the strongest genetic risk factor for AD. And ApoE2 has been shown to generate the highest protective effect against this disease. And so they work in different ways on how they, for example, how this is a risky gene that it exacerbates the amyloid protein aggregation, hinders its clearance, and contributes to the abnormal tau protein activities. So this is one of the most exciting findings. In principle, if one can, for example, convert the ApoE4 gene to ApoE2 gene, then in principle, it should have a very potent therapeutic effect. And advances in gene editing makes it possible to do such a conversion. And so that is the theme of the rest of my talk. Now, this is still an early phase of the work, but I would love to share with you what we have done so far. Many of you probably know this field, gene editing is the most exciting scientific breakthrough in the past decade. Rarely would a Nobel Prize be given in such a short time. This is the second time in the history that the prize would be given within a decade after the discovery. The first one was the iPSC by Yamanaka, and the second one is this one. This was first published in 2013, but within a decade, the Nobel Prize is given. It begins with an enzyme, Cas9 enzyme. It's an endonuclease that would cut the DNA backbone. In this enzyme, there's an epitope that would bind RNA, about 100 base pair or so, or 80 base pairs would be constant. So that leave room for about 21 to 22 bases where one could vary. And then this could form the guide RNA. That this guide RNA binds to this enzyme, forms a complex, and then this complex would then be able to search the genome according to the sequence of the guide RNA. When it finds the right match, it will attach to that site, and then the enzyme could cut at that locus. So that would lead to the deletion of that gene. If then a foreign gene happens to be also in the nucleus, then based on the basic DNA repair mechanism, then that foreign gene could be inserted into the site. So it's a very powerful way to do gene correction. One could delete the mutated gene and insert the correct gene copy into that site, so that it makes in vivo, so that it makes correction of genetic disorder possible now. The biggest challenge to fully realize the potential of gene editing right now is really an effective in vivo delivery system. So in the context of doing, this is a little bit blocked, may not be seen very clearly. in the context of what I was sharing with you, that we would like to explore the use of gene editing to tackle Alzheimer's disease, is that we have to do gene editing of the brain. And that is particularly difficult because of the blood-brain barrier, which has a tight junction, really blocking access of macromolecular therapeutic cargo. And so this is a significant barrier right now because of the BBB. Now, people have been trying to bypass the BBB when they need to deliver biologics to the brain, for example. One can do intrathecal injection, or one could do intracranial injection by opening the skull and to do direct injection into the brain. But these two procedures are highly invasive, and intracranial injection is not really amenable to repeat administration. So we and our colleagues, mostly led by Professor Conofagu at Columbia University, have pioneered the technique of using focused ultrasound to transiently open the blood-brain barrier to allow entry of macromolecular therapeutic cargoes into the brain. In the presence of microbubbles, it will cause cavitation, and that mechanical force would loosen up the tight junction. It is a transient opening of the BBB. So if you have not read this before, you might feel, wow, this seems very drastic. But it is really an FDA approval right now for human applications. So this is just showing in the presence of the ultrasonic energy, the microbubbles can cause cavitation. This is a clinical trial led by Professor Conofagu at the Columbia University. Is on treating patients with AD. Now, this clinical trial is mostly about safety, without any drugs. Just the use of FUS to kind of drain the fluid from the brain, and also as a way to stimulate the immune cells to infiltrate into the brain. In a very early study, she was able to see a modest clinical benefit by this method. And this is, again, as I said, a clinically approved protocol that can be applied in a repeated manner as long as the FUS procedure is spaced by a week or so. But it is a FDA approved method. And this is the study they have shown. This is back to mice right now, that by changing the FUS parameters, one could open up different region and volume of the brain. And that there will be information, but this information is temporary and manageable. And this shows that the BBB will close back within 2 to 3 days, and that there is no long-term tissue damage. So then it gives us the technology to deliver the gene editing elements to the brain to explore this approach. And we use this genetic mouse model pioneered by Jackson Lab. And this has a stop codon. All you need to know is that if we cut this codon, cut this gene, then there will be a restoration of the signal, which will show up in red. And so this is a way to visualize the gene editing if a gene is cut. So this is a study showing, indeed, when we deliver the gene editing element, in this case, it's an all-in-one plasmid DNA with the guide RNA coding for that stop codon. And this region showing up in red is the volume of the brain that has been gene edited. It's a fairly large volume of the brain. And we show that 10 to 15% of the cells in the hemisphere of the brain has been gene edited, because FUS is very focal, local. And so we can use the contralateral hemisphere as the control. And we look at different sections of the plane and so on. So we can clearly see gene editing happening where the FUS is applied. And then we also show that in this region where the cells were edited, that these were neurons. One particular feature of this study was funded by NIH, and it is mandated by NIH that our results can be independently validated by another lab funded by NIH. So Jackson Lab sent their scientists to our lab, watch how we did it. We did, and we got the sample, and we would send the sample back to them, and they would analyze. So their analysis match ours very well. That in the regions of interest, we can get 20 to 30% of the neurons gene edited. This is the first time then one could see an effective gene editing of the brain mediated by intravenous non-invasive method. Now, we make sure that this gene editing technology is robust. We tested in another genetic mouse model, Jackson Lab. The principles are very similar. This is called traffic light receptor model. Again, when the cells were gene edited, one would see the signal in red. So this study shows that in two different genetic mice models, validated in two different labs independently, confirm the robustness of this FUS technology in enabling gene editing of the brain. And this was the first paper published in the field. Now, this was done with adeno-associated virus. So it is the most potent vector for doing gene therapy or doing gene editing right now. But there will always be concerns about the toxicity and the immunogenicity of these viral vectors. So the future for such gene editing is really non-viral. And many of us in the field are working on that. So let me share with you what we have done on this non-viral gene editing. Over the past 25 years, we have been working on non-viral gene delivery using polycations to condense plasmid DNA or mRNA into nanoparticles to deliver into cells. We have also used inorganic nanoparticles to do it. And for this particular study, we focus on this lipid nanoparticle. And the reason is because we pick on this nanoparticle because of this tremendous success of this type of nanoparticle in formulating the COVID vaccine. And we use lipid components that are very similar to the lipid nanoparticles formulated for delivering the COVID vaccine. In essence, there are 4 lipids. The most important component is this ionic lipid. And the other reason is that this kind of lipid nanoparticle can deliver either the messenger RNA or the plasmid DNA, and also it's possible to make the nanoparticles to be small, which is crucial because we need them to go through the blood-brain barrier, even in the presence of the focused ultrasound. So we focus our effort on this kind of lipid nanoparticles. We made about over 1,000 formulations by varying the ratios of those 5 lipids. And then we did high throughput screening in cell culture. We did this high throughput screening a little bit different from the rest of the field. That is, we would like to find lipid formulations that could transfect neuronal cell type well, but to transfect the hepatic cells not so well, because we really don't want the off-targeting gene editing effect in the liver. When we give this lipid nanoparticles, the majority of them will go to the liver. So in this screening, we were really screening for the preferential transfection of the composition on neuronal cells over hepatic cells. And so with this screening, we were able to identify candidates around here that could satisfy our need. Now, in vitro, in vivo correlation is not always that good. So we also did the optimization a little bit differently. First in vitro, and then we will do in vivo, but by intracranial injection to bypass the BBB. And we do the screening by a cluster approach instead of one formulation at a time, because this is not practical in vivo. And after this, then we will go to the eventual evaluation, maybe 2 or at the most 3 best candidates. So we are only beginning to do this right now. But I can show you what we have done in here with the intracranial injection of these nanoparticles. We lower the needle layer by layer, layer by layer, and one could see also a fairly effective gene editing of the brain by these non-viral vectors. And we also show that the cells edited, in this case, a good percentage of them come from the astrocytes, which is very important because astrocyte and microglia would be the most relevant cell types for gene editing, because they are the cell types that secrete the ApoE4 protein. We have done a lot of work to identify the optimal guide RNA, and in pilot study, we could see the knockdown of the ApoE4 gene by this method. So this is where we are. I am just hoping to give you a flavor and maybe hopefully to stimulate your interest in this area. So right now, We are waiting for the funding, hopefully that we can get the support to go deep, to look at both the mirroring study and the mechanistic study on how that works. But mostly is to focus on asking the question, because in this case, we still have to use the viral vector. Focus on asking the question, can ApoE targeted gene editing really achieve the therapeutic goals of delaying or preventing the AD dementia? And so this will teach us something. If it works, then it will aspire, or this will inspire the field, and we can really spend significant effort to develop safe and effective non-viral system. And so that is our goal right now. So very quickly, in summary, what we have shown that because the current drug treatment is so ineffective, it really makes sense that we look at a new approach. And gene editing may be one of the most promising and exciting approaches because it can convert the pathogenic ApoE4 gene to the protective ApoE2 gene. And this is one of the most promising targets. We have shown that the FUS-mediated delivery is a robust technology that could bring the gene editing elements into the brain. And we have also been spending significant effort to identify the promising lipid nanoparticles to do non-viral gene editing of the brain. Hopefully in a few years, we would be able to define the merit of using gene editing to tackle this intractable neurodegenerative disorder. So with that, I would like to thank all my students, fellows, and also the collaborators at Columbia University. This is Hudson River, New York City. This is Broadway. You want to go all down all the way. Times Square, Wall Street, Central Square, and the Columbia University campus is here. It may not be as beautiful as the University of Macau campus, which is truly beautiful, but I still would be happy to host any of you if you would like to come to visit. With that, thank you, and I would be happy to answer any questions if there is time.